Here from this video we will discuss about amyloidosis. Amyloidosis means deposition of amyloid proteins in tissues and organs. Proteins can deposited in various tissues and organs causing variety of problems. Here you can see amyloidosis in a kidney. Actually amyloidosis those amyloid proteins are extracellular, homogeneous and eosinophilic material, and it causes no inflammation in the tissues. Here in this macroscopic view of kidney, due to deposition of amyloid proteins, you can see kidney has become pale in color. Pale in color and kidneys are small because of pressure atrophy caused by the amyloid proteins. You cannot see well demarcated corticomedullary junctions. Here, corticomedullary demarcations have been lost here. The kidneys are pale and small. When amyloid protein is deposited in the kidney, usually sometimes kidney may be normal in size, sometimes they may be enlarged, and sometimes they may be small due to pressure atrophy. Here is a microscopic view of kidney which has amyloid has been deposited. Here there is a special stain called Congo red stain. It has been used in this histological section. From this Congo red stain, this amyloid protein shows salmon pink or orange, light orange in color appearance. We can use H&E staining to detect these amyloid depositions, but in H&E section it is deposited like eosinophilic, then homogeneous and extracellular deposits. Here eosinophilic, extracellular, homogeneous deposits, but the problem is in hyaline degeneration also there is homogeneous eosinophilic material. So we cannot exactly say whether it is due to amyloid deposition or whether it is due to hyaline degeneration. Therefore, in such situations, to confirm amyloidosis or to differentiate amyloidosis from hyaline degeneration, we use Congo red stain or Congo red stain. That is a specific stain. So as you can see here from the Congo red stain, amyloid deposits are seen in salmon pink or light orange in color. To confirm that, we can look under polarized light. If it show apple green bifringence, then it is due to amyloidosis. Here is a amyloid deposition in cardiac muscle cells. Here, the stain which has been used is Lugol's iodine. Here you can see the thickness of the myocardium has also increased. So when we talk about the spleen, when there is amyloid deposition, there is splenomegaly. There are two patterns by which amyloid deposition can demonstrated. There is a pattern called sargospleen and another pattern called lardaceous spleen. Here, this is a normal spleen, this is a sargospleen, and this is a lardaceous spleen. This is a normal spleen, then sargospleen, lardaceous spleen. Here you can see whitish spots are there. These whitish spots are due to amyloid deposition. Here in sargospleen, amyloid deposition take place only in splenic follicles. Here whitish color material is amyloid protein and they are deposited only in splenic follicles. These amyloid proteins are shown in whitish dots here. This is sargospleen. And it is only moderately enlarged and dotted like structure. Then here, this is a lardaceous spleen. Here, amyloids are deposited in the splenic sinuses and vessels in the red pulp. There is more diffuse involvement of the spleen here. It has more enlarged than sargospleen. And here you can see Only intact spleen. There are only purple, small amount of purple dots. That means small amount of purple dots means small amount of intact spleen. Whole whitish color material is amyloid protein. Therefore, it has more diffuse involvement in the lardaceous spleen. Here you can see macroscopic appearance of a spleen with amyloid deposition. Here is a histological section of liver in amyloidosis. Here amyloid depositions are there. Here light pink color amyloid depositions. Here there are normal hepatocytes. Here these are normal hepatocytes. Initially these amyloidosis deposited in space of decay, but then they are deposited in the adjacent hepatic parenchyma and sinusoids as well. In amyloidosis, there can be hepatomegaly as well as pressure atrophy can be seen. The pressure atrophy in this normal parenchyma due to this amyloid deposition. Here is a liver with amyloid deposition. Here again the liver. Here is a macroscopic view of heart after amyloid deposition. You can see the myocardium has been thickened. There are focal subendocardial accumulations as well. Here also a microscopic view of heart after amyloid deposition. You can see pinkish homogeneous eosinophilic material there and normal cardiac muscle cells here. So that's all for amyloidosis, and thank you for watching.